everybody for coming to a new RIDGE webinar from the RIDGE webinar series that we have been organizing, and that you can see the previous ones on the webpage. And it's my great pleasure to introduce you Chong Chen from JAMSTEC. Chong Chen is a deep sea biologist. He received a PhD in zoology from the University of Oxford in England, and his research is mainly focused on evolution and adaptation of organisms to extreme environments, and he works primarily with deep sea vent mollusks. His research interests lies in adaptation, evolution, biogeography, ecology, biodiversity, taxonomy, and systematics. And he participated in more than 20 cruises, mainly in the Northwest Pacific, Descartes Basin, and Indian Ocean. And he has more than 80 papers, and he was one of the lead authors that described the scaly foot gastropod Chrysomalus squamiferum, which is a very interesting name. And without further delay, I will give him the word to talk about the discoveries from Indian and Southern Oceans, new vents, new animals, and new evolutionary pathways. Chong Chen, thank you so much for being with us. And you have the floor.
Thank you very much, Anna, for your introduction. And it's a great pleasure to be here to talk to you all about Indian Ocean and Southern Ocean, well, mostly Indian Ocean today. So I will be talking about new hydrothermal vents, new animals, and new evolutionary adaptations we discovered from these areas in the past few years. And to start off with, I'll be talking about the vents themselves. And to this audience, I think I don't need to introduce to you what hydrothermal vents are, but just to start with, over 20 years has passed since the confirmation of the first hydrothermal vent in Indian Ocean. And so since then, how many deep sea high temperature vents? And today I'm only talking about those ones with vent endemic animal communities, because I'm a biologist, so that's the one that interests me most, have been found. So let's flip back and retrace the history of vent exploration in Indian and Southern Ocean. So this is a map of the main areas we'll be talking about. So if you follow my mouse, then this is the Central Indian Ridge, Southwest Indian Ridge, Southeast Indian Ridge, the CARLSBERG Ridge, and the East Scotia Ridge. So let's start off with the Central Indian Ridge, where it all started. In 1993, the Japanese research vessel Hakomaru conducted plume surveys over the Central Indian Ridge and found plume signals. So this was the cruise, and these are the participants, and they used a CTD Toyo and painstakingly found signals for the Kairai hydrothermal vent field, which was first dived with an ROV, the ROV Kaito from JAMSTEC, on 2000. And it was first discovered on the cruise KR0005, and you can see the participants there. They're all very happy because they found a vent. And then quickly, this was followed up by a cruise from the US on the RV Nor and RV Jason. And they revisited the Kairai vent field, and they found this crazy snail with an iron body and iron scales. And no other animal to date has iron as part of its skeleton. So this was a big news, and it was on Science. But they also found a new vent called Edmund Vent Field, which is just north. So this is Kairai, and this is Edmund. And this was also published in Science. So now we have two vent fields. And then at the end, with the research vessel Hakomaru in JAMSTEC, they found two more plume signals in 2006. And this was an interesting AUV that they took. And then these signals were visited in 2009 with research vessel Yokosuka taking the man-submersible Shinkai 6500, and they found the solitaire hydrothermal vent field and the dodo hydrothermal vent field. And in solitaire, this is a picture of the solitaire in the middle, and you can see lots of life. And especially this white cluster is actually a population of a scalyfoot snail, but it looks white. So remember that, I'll come back to that later. Now, the dodo vent field is the one on the right, and is much less active and has very little animals. And we know that it is no longer active now because on a later cruise, it was actually found to be dead. So now we have 4. Well, Dodo is dead, but we count it anyway. And then in 2019, there was a paper by Ryu et al., which reported the Onuri site north of Solitaire. And this was based on an earlier Korean research vessel, research cruise, which used CTD toyos to detect further signals in the Central Indian Ridge. And then they reported vent-stalked barnacles and Phimorhynchus snails on this picture, which this was followed up last year by a Kim et al. Paper, which formally reported the site with 21 species of animals. So now we have the 5th one. And in 2017, the German BGR discovered two more sites, Alpha and EGS, and those were north of Kyrie Field and very near to Edmund. So they could be maybe considered the same field, actually. And so we've got more. So this is all the activity that has been done in Central Indian Ridge, where it all started. And then the next plume signal was actually detected in the Southeast Indian Ridge. And that was detected in 1996 on ledge 6 of research vessel Melville expedition. And it's called Site 21, and it's located here. So it's quite a distance away from Kyrie. So it's nowhere near where the other vents have been found. And they found this stalked barnacle, which was actually only discovered, which was only formally described in 2018. And it's Neolepis maris indica. And it's the same species as the one in Kyrie and Solitaire. But this was actually already collected by dredging in 1996. But this site has actually not been observed or visited by submersible ever since. So this is there. And then in 2017, again, German BGR, they had this project called Index, which discovered 2 more hydrogen vents. One is Pelagia, and this is actually quite close to the triple junction where Kyrie is. And then to further south, there's New Zone. And the fauna of Pelagia has been reported, and it appears quite similar to Kyrie, but the scalyfoot hasn't been formally reported from there yet. So we have 2 more hydrogen vent fields. And this is all we know about Southeast Indian Ridge. We know very little about Southeast Indian Ridge. So next, coming to the Southwest Indian Ridge. In 1997, 6 plumes were detected, which were visited in 2007 to 2009 by a series of crews by the Chinese research vessel Dayang Yihao, which used CTDs to confirm the signals and used tow cameras and AUV to image one of these sites. And this is the first published picture of the hydrogen vent field, which was called Dragon at the time, but later the name was formally changed to Longqi, which means dragonfly in Chinese. And they reported this shell, which was clearly a shell of the scalyfoot snail. So this actually triggered some interest in the UK, which took the RS James Cook to survey the Longqi vent field, led by John Copley, using ROV and sample the site. So this was the first sampling cruise on this site, and indeed we found live scalyfoot snail. So this was the first site on Southwest Indian Ridge. And then later, more Chinese ships using TV grabs and manned submersible Jiaolong, they surveyed additionally 2 active vent fields, the Duanqiao field, which was very close to Longqi, only about 70 kilometers away, and Tianchen, which was much closer. So this is the triple junction where my mouse is, and then this is Tianchen. So that's much closer to the Central Indian Ridge. So we have 3 active vent sites on the Southwest Indian Ridge that we know. And very, very unusually, last year, a paper came out from the French group using the research vessel Pourquoi Pas and the ROV Victor 6000. They discovered actually a serpentinite hosted vent, not a normal sulfide hydrothermal vent, but similar to Lost City in the Atlantic. And they have brucite calcium carbonate chimneys. But this was the first in Indian Ocean. And they must have been very lucky to find this vent field. And there was no animal reported yet, but we don't know what actually lives there. But considering Lost City doesn't have very much animals, it probably doesn't have very much animals, but maybe it does. We wait to see in the future. So this is where the white star is. And then to the north, we have the CARLSBERG Ridge, which was actually started a bit later. In 2003, the plume signals were first detected. And then in 2007, 2009, further water column surveys, they pinpointed 2 vent fields. And only in 2012, it was actually filmed using a tow camera by the Chinese vessel Dayang Yihao. So then the Chinese research projects used the manned submersible Jiaolong to locate and dive on 3 active vent fields to date. And the first one published was 2017 called Wotan. And this is what Wotan looks like. And it's actually dominated by shrimps, but also there's scalyfoot snail and the hairy snail Albiniconcha. And then the last year, 2 vent fields reported. First, the Tianshu vent field. And we can see on the picture that there's high temperature venting dominated by Rimicaris, these blind shrimps that rely also on their symbiotic bacteria living inside their head, but also in the digestive tract. And diffuse flow areas dominated by sea anemone. So this is quite a large area, but apparently mostly inactive sulfide with just some active chimneys. And the last one is the Dashi vent field, which was also published last year. And it's also dominated by these blind shrimps. And the fauna is similar to vents in Central Indian Ridge, but it doesn't have the scalyfoot snail, the hairy snail, the mussels, or the stalk barnacles, as far as we know. So we have 3 vent sites on the CARLSBERG Ridge. And then coming to the Scotia Sea, the Scotia Ridge, which is here. In 1999, the plume signals were found over 2 segments of the East Scotia Ridge, one in the north called E2 and one in the south called E9. And this was pinpointed by a UK research cruise with the James Clark Ross icebreaker, and that used CTD tow wheels and tow camera to confirm the presence. And then there were 3 crews with the RS James Cook and the ROV Isis, and also ROV, and they confirmed the vent fields and sampled there. So this picture is quite famous with all the Kiwa Tyleri crabs. So we have 2 vent sites. And then there is also another one to the close vicinity of E9 in a collapsed caldera called the Kemp caldera. And this vent site is very different from E2 and E9 in that there were large areas of sedimented area dominated by these Chaetogena-like clams, which we know now are Archivesica protodesiadoi, which was originally only known from Argentina. And then this site was revisited in 2019 by the German icebreaker Polarstern. So this area hasn't been visited very much at all. So now we have 3 vent sites here. And actually, this isn't the only vent sites in the area. There is actually 2 more sites that we know known from 1995. And in 1997, they were sampled, but only with dredges. And then this was never seen with large, dense vent animals. And in Bransfield Strait only, so these chimneys are known. There's bacteria mats, but there's no dense vent fauna. But then when in 2011, they were revisited by the UK's RAS James Cook, they actually managed to collect tube worms, colonies of very, very thin tube worms. Unlike those like giant tube worms, they're very large. They're closely related, but they're hair thin. And they're Sterolinum cornutum tube worms. And they were actually surprisingly conspecific with those from Arctic and Gulf of Mexico. So this is a cosmopolitan species that has populations both in Arctic vents and the Southern Ocean vents. And probably the same species was also found in the Kemp caldera. So although not densely populated by animals, this Bransfield Strait sites do have animal communities related to hydrothermal vent activity, because these worms also have endosymbiotic bacteria, which they rely on. So without hydrogen sulfide, they will not be able to survive. So these two sites are here. And lastly, there are sites that were discovered in the last decade on the Australian Antarctic Ridge. And in 2011, 2013, there's a Korean research vessel, Aron, which conducted plume surveys, and they found the Mujing vent field, which is south of New Zealand, actually. So on the right other side. And vent animals, including kiwa and these 7-armed sea stars, which were previously only known from the East Scotia Ridge, were actually found in this site. But this site has also not been visited by submersibles, either ROVs or manned submersible to date. So much more is to be learned from this area. So this is actually here on the map, on the right-hand corner, this star. But obviously, if we look at the map, we have these empty areas. We have this empty area around Andaman, and we have this empty area in Red Sea. We have this empty area around the Bouvet Triple Junction, and we have this area on the southeast Indian Ridge. And even this Site 21 has not been visited by submersibles. So there are a lot of empty spaces to fill in terms of understanding vent biogeography and the evolution of vent communities. And then if you look at the Interridge Vent database, we actually have active signals of active vents in all these areas, but they just have not been pinpointed or visited by research vessels using submersibles. So we really need to do that if we want to understand the vent biology and also geology and all the sciences in this area. And another interesting point to make here is there are actually seeps of India that were reported recently, first in 2019, with lots of animals, and also again in 2020, which reported even more sites, so just east of India. So these animals seem to make a connection between the Pacific and the Indian Ocean. So there might actually be interesting things to discover in the eastern part of Indian Ocean in the years to come. So these are here. So maybe events here will be interesting too. Okay, moving on. So next, we're talking about new animals. Well, lots of animals were actually already described in the early 2000s when the Tyre vent field was discovered. So such as the shrimp Primotyrus tyre and the small snail Disperageomus indita and Bruciella vareni, Ephinopsis, and more vent shrimps, vent crabs, and the mussel. These were already described in the early 2000s. But actually, many animals which we knew were there were only described in the last few years, by which I mean after 2015. So even the scalyfoot snail was not described and named until 2015. And many are still undescribed that we knew to be there from ages ago. So these pictures, most of these are actually from the Vandover 2001 paper in Science from the Tyre vent field. So these have not been described for over 20 years. But some progress has been making recently. So the Korean groups have been very actively describing new species they found in the Central Indian Ridge sites. And for example, we have a surprising discovery of a new chemosymbiotic mussel species that eluded the previous search, Gigantidus varani, and which was never actually described from, never found in any other sites. So there are still many more to be found. And also we have the first taxonomy papers of the vent copepods, which fills a very important niche because myofauna has long been an unknown area in Indian Ocean and Southern Ocean, like there has been no data. The first data available for myofauna was actually by Zabina Dorna in 2016 in PLOS ONE, where she did, her group did some work on barcoding and the genetic diversity of lots of copepods from around the world. in vents. And then they found 2 new species of Stygiopontius in the vent-specific family, Derivotidae, from the Central Indian Ridge. And only in 2020, finally, 9 species of tropopods were described from hydrothermal vents in the Indian Ocean, mostly in Onuri, but also in solitaire by Taurian groups. And the only issue with these is they do not include barcoding data, so we actually don't know which species they described are maybe the same as the ones the Vine found or not. So that's a problem we need to solve in the future, but at least the meiofauna is starting to move forward. So what I want to say is, looking at the species list we had from various papers, how many species have we already named and how many have we not named? So this is the taxonomy chart from Cindy Van Dover's 2001 paper in Science for Kyrie and Edmund Benfield. So if we look at this, these animals that they found in Kyrie, the ones with the white background, such as Bruciella, Dysprasia, Austingrea, Rimetaris shrimps, as I said, they have been described in the early 2000s. And there are some species which we don't know what they refer to, or they have not been recollected. So these we will ignore. And the one with the yellow background are actually still undescribed. And those with blue background are only described in the last few years. So just looking at this, we have actually about half of the species that were found in 2001 that are still not described. So there's a huge taxonomic lag in the Indian Ocean and in the Indian Ocean vents. And if we look at an updated list in Watanabe and BDC 2015, and we see a similar picture. We see that about like a third of species have been described in early 2000s, and about a third-ish have been described in the last few years, but lots more of work to do. And this is similar in Southwest Indian Ridge, where because these vents share a lot of species with Central Indian Ridge, so some of these are described in early 2000s with the white background, but we have, although we have been working really hard in the last few years, and so these species mostly have been described after 2018, but there are still many species that don't have names. And then this is Chen vent field, which is the newest vent field we found from Southwest Indian Ridge, and we also have undescribed species from here. And these species are actually not all new species that were found from Chen. Like, for example, Euleptopsis have already been known from Kyrie since 2001, but nobody has described this. And moving on to the Antarctic vents. Similar, because Antarctic vents are relatively new, and they were not described, they were not discovered until 10 years after, they were not found 10 years after the Indian Ocean ones. So many species were actually described in the past 5 years. And the situation is actually great for some groups. For example, in the mollusks, we actually finished describing all the new species of mollusks last year. So that's done. And most of the arthropods are done. But we have groups like the Cnidaria, the sponges, the annelids, that have completely not been worked on at all. So there is still a taxonomic lag in East Rochelle Ridge. And this is a similar chart from Kemp Caldera. And this table shows you also have the similar problem. So the sponges, the sea anemones, the polychaetes are the ones that are really lacking effort in the East Rochelle Ridge and in the Antarctic vents, and needs to be catched up with groups like mollusks and crustaceans. And if we look at following similarities, and then there is actually a clustering based on the geographic region, and also based on the ridges. but we still lack the CARLSBERG ridge data. So we do see some similarities and differences among the vent fields in terms of composition based on their geographic distances. But coming to looking at connectivity properly, we actually don't have much data. So the scalyfoot snail was the only species with connectivity study across two ridges in the Indian Ocean. That is between Southwest Indian Ridge and Central Indian Ridge until 2020. So this was a paper published by myself in 2015, and it used one single gene, the CO1 gene, to look at the connectivity between sites in these two ridges. And the black and the gray are sites in Central Indian Ridge, and the white is the single site, Longqi, in the Southwest Indian Ridge. And as you can see, there are not very many shared haplotypes between Southwest Indian Ridge and Central Indian Ridge, actually only one. So at the time, we inferred that there was a lack of connectivity across the ridge between Kairay and Longqi. So we thought maybe the triple junction where the two ridges change was where the larval dispersal was stopped. But recently we found the Tianchen vent field, which is much closer in distance to Kairay. So we could actually test whether this was true. And we did, similarly, we used the CO1 gene, and we did connectivity studies across these sites using 3 species, the vent crab, the mussel, and the snail, the scalyfoot snail. And the mussel has a very high dispersal capability. We knew that from before. And so it's not a surprise that for the mussel, they are basically everywhere, and they are well connected across the entire region we studied. But for both crab and the snail, which should have much less dispersal capability, they actually showed a lack of connectivity, not between the two ridges, but they were well connected between Kairay on the Central Indian Ridge and Tianchen on the Southwest Indian Ridge. Instead, there was a lack of connectivity between Tianchen and Longqi on the single ridge, on the Southwest Indian Ridge. So it is not here where the connectivity broke, but instead it is here. And if we look at the map, we can see a lot of transformation faults. And we think now that this is what is stopping the larvae from dispersing between these two areas, which makes Longqi and Duanqiao very special regions in terms of biogeography. And in the future, it would be great if we can take further studies with species such as Neolepis mercinita, the stout barnacle, which we know they are present across the entire area, from CARLSBERG Ridge to Longqi and to even Southeast Indian Ridge. So this would be, for example, a great target species for the future. And in terms of East Scotia Antarctic vents, there has been some studies on the connectivity between East Scotia Ridge sites, and the 2 northern and southern sites on the East Scotia Ridge are actually very well connected, whereas the Kemp caldera is not connected with the 2 sites on the ridge. So that suggests a local retention within the caldera-based Kemp caldera. But otherwise, on the ridge, the 2 sites are well connected. Right, so let's talk about the really exciting part, which is the adaptations of the animals, specifically the 2 animals that I've been studying over the past few years, the scalyfoot snail on the left and the Jackson pelter snail on the right. So these scalyfoot snails, they live under the shrimps, and they're very hard to see and very hard to collect. But once you see them, they are shiny. They're shiny like metals. And actually, since this discovery, it was quickly realized that they are actually also chemosymbiotic. So they have these specialized gland inside their, in human, it would be like their throat. So in the esophagus, they have this huge pouch, which is filled with bacteriocytes housing a single sulfur oxidizing bacteria in their trophosome. And they also have a very large circulatory system, which we think they use to suck out the chemicals from the vent fluid to supply the bacteria inside their throat. And they also have one of the largest heart by relative volume among living animals, which they use to pump blood with the chemicals in to supply the bacteria. But most people, such as like Anna just said, know it for this iron scales. And it is indeed the only snail with dermal scales, which grow in an imbricating manner. And in Kyrie field, the scales and shell are mineralized with iron sulfide. So here we have a cross-section of the scale, and you can see these black bits inside the scales cross-section, which are actually particles of iron sulfide, mainly pyrite and graphite. So graphite is actually magnetic, like magnetite, but only with sulfur. So this snail actually sticks to magnet. But later we also found this solitaire vent field, and the snails living there are actually white and without iron, but genetics show that they're entirely the same species. So why is the scales black in Kyrie? So is the iron sulfide formed by passive deposit, or is it active production? Is it made by surface bacteria living on the snails, or is the snail making this itself? Where is the iron and sulfur coming from? And what are they using the scales for? Is it really defense? Now, here's a video of me playing with the snails and a magnet. So they do stick pretty strong to the magnets. So studies were mostly done when only the specimens were known from Kyrie, so only the black ones. So people assumed that was normal. But then solitaire specimens have no iron sulfide. So in the middle, we have the scale from solitaire, which is entirely white. And then left, there is one from Kyrie. You can see the difference. And then we found this Longqi vent field, which actually have a gray scale, which is somewhere in the middle. And that seems to vary with the environment. And it actually correlates with the iron concentration in the vent end member fluid, where the Kyrie vent end member fluid is actually 58 times richer in iron than solitaire. But sulfur is consistently present in all three populations, regardless of iron. So that led us to question whether the sulfur and iron actually come together. So we measured sulfur isotope ratios of pyrite in the scales versus other materials from Kyrie vent field, including the soft body of the snail, the foot, basically, and then the chimney minerals, and also the vent fluid, and the body of a vent muscle. And we found that actually the scales are much lower. They had a very different delta 34S from the natural minerals. And this usually occurs when there is biomineralization. So the animal is preferentially accumulating isotope of the sulfur through some sort of biomineralization. Whether it's bacteria or animal, we can't tell from this, but it seems to be some sort of biomineralization. And then we look at the scale itself through electron microscopy. So we took cross sections of the scales and mapped elements on it. And we found these circular elements. And these circular elements, if we look at the panel B, so these bright white ones are actually iron sulfide. So they have both iron and sulfur. But in these gray circles, there's only sulfur, but no iron. So it seems that iron and sulfur are not always formed together. And this implies there's different source of sulfur and iron coming together, as we suspected. And to pinpoint what is happening, we took a section of the scale. So the scales is actually here, and you can see the tissue in the middle. and that's the tissue secreting it. And actually, there's less sulfur on the inside, so the tissue, but more sulfur on the scale, the newly formed scale. And whereas iron, there's actually more iron on the inside, and there's no iron on the newly formed part of the scale. So sulfur is deposited actively by the snail from inside of its body out, whereas iron is not. So, which implies the iron comes from outside. And to check this, we actually did an in-situ experiment where we planted white scales from solitaire for 2 weeks in the Kyrie vent field where the black snails naturally occur. And we found the scales. So the panel B and C on the upper right-hand corner, they're the same scales. So before and after 2 weeks in Kyrie vent field. And they actually became black and with iron sulfide. So this showed that the iron indeed is actually coming from outside. And then we did some 3D modeling of the internal structure of the scales. And each of these columns is approximately 50 nanometers. And you can see that there are lots and lots of pillars. And these pillars are actually columns of iron sulfide in the black scaly foot. So the columnar structures extends in a linear fashion from the secreting cells upwards towards the snail, towards the scale's tip. So basically what we found is from the cells, the animal is actually only excreting sulfur. And the iron ions from the vent fluid diffuse in and reacts with these sulfur columns. And then that turns the sulfur into iron sulfide. And this is why in the iron poor vents, there is no iron and only sulfur. And the sulfur in the scales originate from the snail, which are extracted from the vent fluid, transported by the animal to the symbiont, used by the animal symbiont, and then transported again by the animal to be deposited in the scale. So we think it is a removal mechanism, a detox mechanism of use excess sulfur from the animal outside of its body. And it's an adaptation for symbiosis and actually not protection. And so where did the scale actually come from? So among living mollusks, so close relative of snails, there's only groups called Aglyphera, chitons, so like this mollusk with 8 shells. So these guys have scales, but the snails do not. So none of the snails have them, but some of their close relatives do actually have scales. But once we look at the construction, the scales are completely different. So in the scaly foot, these are proteinaceous and chitinous structures similar to our fingernails, and they grow in a similar manner to our fingernails actually, in layers. Whereas in the chitons, although they may look similar, they are actually, the scales are made by pouring calcium carbonate into a pocket, and then they're solidified. And so they are calcium carbonate structures. So they're very different, although they may look similar. And they are actually more similar to scales of Cambrian animals that we only know from fossils that also had a similar scale with a core, which supposedly deposited the scales. But of course, since the scaly foot, the entire lineage containing the scaly foot only dates to late Jurassic at most, they can't be homologous with the structures that were made by these Cambrian animals. So how did the scales actually evolve? And originally, because snails often have operculums to close their aperture, so it was originally suggested in the original paper that first reported the scaly foot's existence that this is probably a duplication of the operculum. But later, it was found that the scalyfoot actually does have a operculum, and which looks completely different from the scales, and they have different types of secretory cells, which implies that they are actually a novel structure and not homologous to the operculum either. So you can see here, they had a true operculum when they are small, and they also have a true operculum when they're adult. It's just difficult to see. Right. So what we did was we tried to trace the origin of the scales using genomic methods. So we used whole genome assembly. So we sequenced the scalyfoot genome using nanopore, Illumina, and high-C sequencing to obtain a chromosome-level whole genome assembly. And this is actually among the highest quality genomes so far among mollusks, even today. And we also had transcriptome data to look at the gene expression from specimens fixed in situ in RNA preserving liquid, so that prevents the RNA from changing between collection and coming up on board through a different temperature and pressure gradient. So the gene expression data we get is as close to natural as possible for these deep sea animals. And what we found is there is actually a very highly expressed series of genes only in the scales and not in other tissues. So looking at the literature and comparing with other closely related animals that produce hard parts, we realized that the scale production is mediated by the same genes, transcription factors, that control the production of hard parts in closely related groups of animals called ophiutrochozoans, which includes other mollusks, of course, and also brachiopods and also polychaete worms, which produce various types of hard parts like chaetae, shell, and scales, and so on. And so this biomediation toolkit is actually shared by all these lineages. So what this means is the toolkit, the genome, the genes for making hard parts is the same across the shell, the operculum, and the scales, but they are actually different evolutionary events that led to these hard parts arise entirely new, but from the same basis of genes. And so the scale is a novel structure, but it didn't really need to re-evolve all the genes necessary for making the scales because it already had them from Cambrian at least. And this is what we call a deep homology. So we confirmed the deep homology between the scalyfoot scales and hard parts that are known from other ophiutrochozoans. So it's actually wasn't that hard for the scalyfoot to make the scales, as you might think. And also we found that the genes that actually, so what we were talking about the toolkit is actually the upstream, the genes that control the downstream genes that actually do the secretion. So we found that the genes that do the secretion are often the same genes or similar genes that were co-opted from production of shells. So they use the same genes or similar genes to make shells and also to actually make the scales, but they are controlled by a different manner within the same toolkit. But still, we have lots of things that we don't know, because as is with all kinds of genomes in these minor groups, we have 11% of genes that we don't know what they do in the scalyfoot snail, and it will be a long time before we can actually understand how these upstream genes interact with downstream genes and to make the scale. But what we know is that the control mechanism for the scales is actually ancient. Now, let's talk a little bit about Gymnopelta. So Gymnopelta is also in the same family as the scalyfoot snail, and there are two species. One is Gymnopelta chessoia, which lives on the East Scotia Ridge, and one is Gymnopelta aegis, which lives on the Southwest Indian Ridge. And they're like scalyfoot, they're much larger than other members of the family, and that is because, as you can see, when you have these huge, dense aggregations of animals of large size in hydrothermal vents, you would think, they probably have symbounds, and that is true. And they also have endosymbounds, like the Stalyfoot, they have a pocket in their esophagus, which they house endosymbounds. And the interesting thing is we found they are actually not the most closely related species in the group. So this leads us to think that they probably are convergent in having endosymbiosis. So endosymbiosis probably evolved twice in this one family and not just once. And when we look at the anatomy using synchrotron micro CT, where this really fancy synchrotron we use to make, we use the X-ray emitted from synchrotron to image the internal anatomy of these snails instead of actually having to dissect them. And we found there's a nutritional shift between about 2 to 5 millimeters in size in Jarenpelta. So between about 5 millimeters to 8 millimeters, they switch from normal grazing, eating bacterial film to having endosymbiotic bacteria. And this is not seen in the Stalyfoot and supports the fact that they have different development mechanisms and therefore probably arise separately in evolution. And if we look at endosymbionts, there's no endosymbionts before this 5 millimeter size, and then suddenly there's cells packed with endosymbionts. But what's interesting was that if we look carefully in the TM image that we got from these snails, so these smaller bacteria without the stacking membrane are probably, we thought, are self-oxidizers, which we got predominantly from sitting as sequencing of the barcoding gene. So we knew this was normal, but we also found there are rarer but larger bacteria with stacking membrane, which are typical of methane oxidizers, which we could not detect with sitting as sequencing so far. And then only recently, we were able to use omics methods and actually sequence lots and lots and lots of sequences of this organ. And we could finally pinpoint that there were actually indeed 2 symbionts. And the methane oxidizer is about 10 times as abundant as, only about 10% as abundant as the sulfur oxidizer. And with fluorescent in-situ hybridization, which using specific probes to mark these 2 different lineages, we could see that there are indeed 2 separate symbionts in the cells. And we then sequenced and assembled the genomes of the 2 symbionts and the snail host. And we found that in Jarenpelta, we have 2 symbionts, but they're both gamma proteobacteria. One is sulfur oxidizing, one is methane oxidizing. And also having these genomes allowed us to compare the biosynthesis capabilities of key amino acids and vitamins by both the host and the symbionts with the Stalyfoot snail, for which the genome, as we said, was already published, and also the symbiont of the Stalyfoot, only one symbiont, sulfur oxidizer, was already published by Nasrallah et al. In 2014, so we could make comparisons. And interestingly, we found that even when the snails were quite closely related, the sulfur oxidizing bacteria of the 2 species are actually not closely related at all. They are actually very different bacteria within gamma proteobacteria, but they're totally different symbionts that were picked up by the 2 snail species, which live together side by side. And when the snails couldn't make an amino acid or a vitamin, both of these bacteria, although they're not closely related, were actually able to make these nutritions required by the host snail. So although they're distinct related, they have somehow the same capability to synthesize these key amino acids and vitamins that the host cannot make themselves. So perhaps this is a key criteria for selecting which lineage of bacteria you encounter as your symbiont for these snails that actually cannot feed to give themselves a supply of these amino acids that they don't have. And so that's all for the 3 main topics I want to talk about. And I just want to move on and make some ending remarks. So most of the research cruises and discoveries in Indian Ocean since 2000s have been linked to deep sea mining. And almost all confirmed Indian Ocean vents are now in ISA exploratory license areas by China, Korea, Germany, and India, except Solitaire Field, which is actually within the exclusive economic zone of Mauritius. So how do we ensure conservation and sustainability while balancing with these contracts? So for example, East Asia Ridge doesn't have this problem because it is protected by the Antarctic Treaty and also the South Georgia and the South Sandwich Islands protected area. So it's a very different situation between the Antarctic vents and the Indian Ocean vents. And I was also involved in making a global scale trait database for vent animals. But what we realize is the data we have is very skewed to the areas we studied the most since the discovery of vents in 1977. So mostly East Pacific, and also a lot of data from Mid-Atlantic Ridge, but very little from everywhere else. So as Stella and Ammon said very well in their 2019 paper, we studied the North. So among the Northern and Southern hemispheres, we have a lot more, we have more vent fields in the Southern hemisphere, such as Indian Ocean and Southern Ocean. But we have lots of research crews in the Northern hemisphere and not so much in the Southern hemisphere. And since we know that it's difficult to translate directly the knowledge between the different regions of hydrothermal venting because of different properties, such as spreading rate and also the fluid dynamics, it's difficult for us to assess how vulnerable these Southern hemisphere vents are to potential disturbance such as mining activity. And if we don't have taxonomy and basic biology, we don't have information to make these conservation decisions or measure resilience. And this actually led the scalyfoot snail to be listed as the first endangered species due to risk of deep sea mining on the IUCN Red List. And this listing requires data on life history traits. And we accumulated a lot of data for the scalyfoot through different studies because it's such an interesting species. And then we now have completed actually 61 assessments published. And a lot of the Indian Ocean species are critically endangered or endangered. And all those least concerned species are actually in protected areas. Oh, by the way, these are all mollusks and we need help with assessment for other groups. So if you're a taxonomist and if you're interested, please let us know. And especially the Indian Ocean is rich in critically endangered species because there are many species that live in only 1 or 2 hydrothermal vents, which are both or all in these mining exploratory areas. And also I would like to advise that in September, there will be a special session in the IUCN Congress, and that is the deep sea red list session. And we were selected from about 1,500 special session proposals. And this is also a virtual event. So if you're interested, please have a Doodle search and look into it. And we are hoping this will lead to a deep sea animal working group in IUCN, so we can actually do more assessments for other animal groups and not just mollusks, because currently we are actually embedded in the mollusk specialist group. So we need a specialist group on our own to work on other taxa such as polychaete worms and crabs and shrimps and so on. Right, so in conclusion, what we need for the future vent biology in this area is we need to sample and visit vents in the missing pieces of the map. And we need more descriptions and naming of species, especially in the minor phyla such as sponges and sea anemone, and these groups that normally there's not very well worked on in terms of hydrothermal vent biodiversity. Because if we don't have names, we can't use them in conservation contexts, such as listing them in IUCN Red List. And we need better connectivity studies in the Indian Ocean. Currently, we only have one gene, so we need better, for example, SNPs level to reveal more intricate details in the connectivity and dispersal capabilities. And also we need more data on generally morphology, life history, and physiology from animals in these areas. And we need to make sure we feed this knowledge and effort into conservation, because the Indian Ocean is very much in need of that effort. And for that, it's very important that we have collaborative sharing of samples available from different sites. There are many recent cruises from, for example, ISA contractors, but there is actually no, well, they have obligation for sharing of data, but we don't know about specimens. So many specimens collected from the physical deep sea quickly sinks into a metaphorical abyss, becoming invisible and available to taxonomists. So if you have samples that you cannot identify, please make sure you share it with taxonomists who can work on them. And to end, I would like to thank my collaborators, who I haven't listed all of them, but yes, I thank them all deeply. And also for the funding agency that funded my research. And thank you very much for your attention. And I'll take any questions.